So welcome to today's webinar titled Improving Single Genome Annotation by Multi-Genome Analysis by Gustavo Gussman. Gustavo is a senior research scientist at the Institute for Systems Biology. He's currently developing data structures, algorithms, software, and visualizations for analyzing whole transcriptomes and genome sequences in the context of cancer and of families affected with a variety of disease phenotypes. Gustavo, you now may begin your presentation.
Okay, thank you. As we all know, we are entering the era of personalized genomics and personalized medicine, and soon enough, everybody's personal genome is going to be part of the medical record. And the question is then, how can we improve the annotation of every single genome? So, as we all know, the acquisition of genomic data is improving, is getting very quick, but actually interpreting the genomes is lagging behind. Sorry. So what we want to discuss today is this idea that we very quickly moved from sequencing the genome to sequencing individuals, families, and all this data is now accumulating. We have population genomes, but in the clinic, whenever you go to see the physician, physician sees only an individual, and you need to interpret that individual's genome. And every genome has plenty of errors, false positives, false negatives. So the big question is, how can we use knowledge gained from analyzing many genomes to improve the interpretation of each individual genome? And I want to claim that doing multi-genome analysis can help significantly for this purpose. So as we see here in the slide, from sequencing an individual genome, we can learn about the genome of the family, and we can also accumulate that information into the genome of a population. And then the big question is, how can we take this kind of information that we gain from accumulating many genomes to improve the annotation of a single genome? This dashed arrow here shows this idea that, as we published a few years ago, when we did the first analysis of genomes in a family, you can take the genomes of 4 people in a family, in this case 2 unaffected parents and 2 children affected with Miller syndrome, and use the combination of all four genomes to interpret, to identify which gene causing the disease. In that case, we use 4 basic constraints to identify the gene causing, the disease causing variants. So, for example, we knew that this is a very rare disease, and we expected the inheritance to be recessive. Since both siblings have the same disease and it's rare, then we expect the gene causing the disease to be in a region where the 2 twins are, the 2 siblings are essentially identical twins. Again, the disease is very rare, so we expect the variants to be very rare or completely novel. And naturally, we expect the variants to be detrimental. So using these 4 constraints, we identified out of the entire genome, we identified just 4 candidate genes, and that was back then. If we were to repeat the same analysis today, we would dismiss 2 of those variants as false positives. So since then, our team has expanded quite a bit the scope of analysis. We moved from 1 family with 4 people and using all 4 constraints that I mentioned. Now we have many studies, including over 900 people, and consequently, we definitely had to enhance our ability to analyze many genomes, and we started learning a whole series of additional constraints and concepts that we can use to improve the analysis. So this led to the creation of quite a big pipeline. So this is a part of the pipeline, and the idea here is to develop a series of algorithms and tools to implement all these different kind of concepts and analysis that we developed, ideally going from genomes to interpretation. And as you can see here, we have a whole series of different things that we're needing, all the way from project management to understanding basic biology of the genome, and then sorry, and then comparing to other genomes and trying to interpret the variants themselves. In parallel to this homegrown pipeline that we developed at ISB, We are also using Ingenuity variant analysis, which, as you know, is commercial software that makes it quite convenient to simply upload the genomes, run a whole series of analysis inside the same tool, and then lead to visualization and interpretation of the results. So going back to our own pipeline, the big point I want to make here is that some of these tools are intrinsically multi-genome, so they only have meaning because we are analyzing several genomes at the same time. On the other hand, some of them are intrinsically single genome, but they are particularly informative when we take them in multi-genome context. So for example, one of them is kind of trivially, if you accumulate lots of variants observed in different genomes into one single database, then you gain a lot of information about the frequency of the variants in the population. So Kaviar is a database of known variants that I developed at ISV. And the idea here is everybody who wants to do genome analysis needs to, in some way, download DBSNP and the 1,000 genomes data and NHLBI, several thousand genomes. But in addition to those genomes and exons, there's a very large number of private genomes, personal genomes, that have been released. And there's also lots of information from RNA-seq that also gives data about personal variants. So what we did here is to compile all these different variants into a single database and make it publicly available to reduce the amount of effort that other people need to do to combine all this different information. Another kind of analysis or tools that we created is a tool for annotating functional implications of different variants in the genome. We call this FAVA, for functional annotation of variants. And the idea here is just like other tools like ANNOVAR, you observe one variant, you want to know what that does in the context of the genome. And the approach we took here is one that we think is quite convenient for making the analysis efficient, which is to pre-compute for the entire genome every single position that you might change will not have any kind of effect on a gene, on a protein, or any other kind of functional notation that we can envision. So this is intrinsically not multi-genome, but if you take this kind of genomic analysis in a multi-genome sense, some interesting information comes out. So imagine, for example, that this line here represents schematically all the variants observed in some region of one genome. And those variants annotated in red are those that, according to FAVA, they have some important functional implication. So if you do this for one single genome, this is what you observe, some randomly or apparently random distribution of functionally important variants. If you do that for several genomes, then you get this kind of picture, again, very schematically. And as you look at more and more genomes, at some point you realize that some of these variants are shared by many of them, so they're presumably not that important because many people can have them without many implications. But then there is a number of variants that appear to be detrimental, and they very clearly cluster in certain regions of the genome. So in this case, again, schematically, this stands out as particularly interesting. There are some other clusters that might be potentially interesting that's left to statistics. But in this case, what we do is we identify such clusters of positions that appear to be mutated frequently in different people. And we define this as what we call commonly mutated segments. And these segments are presumably pretty prone to false positives. So we define this resource that we call commonly mutated segments. And everybody who has done any analysis of a personal genome, we know that every single time you find variants in genes like titin or factor receptor genes or mucins, and these are very frequently false positives. So it is very convenient and common to simply dismiss those genes. If you find a variant in titin, just ignore it. But we, of course, know that that's not always the case. Sometimes variants in titin might be important. So the use of this set of commonly mutated segments is to filter out parts of those genes that appear to be error-prone. So here in the figure I show, for example, the MUC6 gene, a mucin gene, that instead of dismissing the entire gene as unreliable because of this frequency of false positives, we can identify that there is one exon here, one very long exon that accumulates lots of variants, as you can see here from the divisionib track in the UCSC browser, large accumulation of variants that appear to be detrimental. In addition to that, there is a couple of other regions inside the gene that appear to be, again, error-prone, so we can dismiss those. But if you were to find a variant in some other exon, for example, in this exon here, then there is much less reason to dismiss that as a probably false positive. So we're talking a lot about identifying errors. So I want to go through a number of different types of errors that we identify by comparing genomes. So the first kind of error is very simple, and it involves only one genome. It's just a very simple no-call. It's a technical error. And it's kind of important to recognize that sometimes we observe a single nucleotide that is no-called. Sometimes we observe patches that are not called. And these two behave in different ways when studying multi-genomes. The second kind of error involves comparing either two genomes, a parent and a child, or comparing three genomes, a trio. And these are of course, Mendelian inheritance errors. And we recognize two types of these. One of them in which the child doesn't appear to have inherited anything from one of the two parents. And the other case in which they seem to have inherited one allele from either of the parents, but then there is a second allele that doesn't seem to come from anywhere. So these two Mendelian inheritance errors, they're of course nothing new, but we could identify by studying a quartet an additional kind of error that is less trivial, requires a bit more explanation. So when we study a quartet, we can identify 4 different states of inheritance in the way we call them. So if the two siblings inherited exactly the same alleles from both parents, we call that the identical inheritance state. If they, on the other hand, inherited opposite alleles from both parents, we call that non-identical. And then there are the two intermediate states in which, for example, both offspring inheriting the same thing from the father but different alleles from the mother. We call that haploidentical paternal. So 4 inheritance states, color-coded here in this way. So inheritance, the identical state called in red, for example. And what we recognize is that different genotypes observed in the 4 people in the family can be consistent with all 4 states. For example, here, if everybody is homozygous for the same thing, that is consistent with all 4 states. On the other hand, some other genotypes may be consistent with only 2 states. So for example, here, if both parents are heterozygous AG, one child is heterozygous AG as well, and the other one is homozygous A, this very intuitively is not consistent with the 2 siblings being identical or non-identical. So we can study the genome of the family as a quartet, and for every position where we recognize a situation like this that gives support to the 2 haploidentical states, we can accumulate this kind of support for the 4 different states along the genome and create from that map that we call ISCA for inheritance state consistency analysis. So this is an ISCA plot for the Miller syndrome family. And we can see here that, for example, here in chromosome 6, there is this large region painted here in red, in which the vast majority of the variants support the idea that 2 children received exactly the same alleles from both parents. Then that transitions through a recombination here into a state in which they receive the same thing from the father, but different things from the mother. So clearly that transition point is a recombination in the mother. And that transitions again through a recombination in the father into another state, here blue, where both offspring receive different alleles from the parents. So having such a map, now we can very intuitively identify a new kind of error. So if, for example, we observe in some position in the genome, again, this same genotype form in which clearly two offspring did not receive the same alleles or opposite alleles. So we say here this supports haploidentical states only. If this is embedded in a region in which the two children are supposed to be identical, then we recognize that this has to be an error. So having all these different types of errors, we can look now at the distribution throughout the genome. And we can see, for example, that when comparing a large number of trios and simplifying the problem by taking only good quality variants outside repeats, so we can see the distribution of Mendelian inheritance errors is really non-uniform throughout the genome. Certain regions accumulate more errors than others. So if you look, for example, at an area here, you can see that there are certain peaks of Mendelian inheritance errors, particularly in dark cytobands. And as we all know, those bands correlate with low GC content sequences. So indeed, we identify that pretty much as expected, there is an accumulation of errors in very low GC content sequences, but also in high GC content sequences. This is pretty much as expected because the more extreme the GC content, the harder it is to map or read uniquely. There's more error in mapping, therefore presumably more error in genotype cloning. We similarly did the same analysis for state consistency errors, and we identify again that there is skewed distribution. Some areas of a genome have more state consistency errors, and this is not exactly the same distribution as with Mendelian inheritance errors. But nevertheless, we see again that state consistency errors again accumulate in low and high GC content regions of a genome. So now we want to switch to a specific analysis of a specific disease. So we have been collaborating with Kathy Swoboda from University of Utah School of Medicine on analysis of alternating hemiplegia of childhood, which is a rare neurological disorder with paralysis on either side of the body, particularly in children. So most cases of AHC are sporadic, but there's also some family cases that Kathy could collect. So we have been sequencing these genomes, the genomes of people in these families, both affected and unaffected people. The sequencing was done by Complete Genomics, funded by University of Luxembourg. And we've been doing the analysis using both our resources at ISB and Ingenuity variant analysis. So when we started this project, we had this set of 18 sporadic cases we sequenced, and also several individuals in 5 different families. All these were sequenced by Complete Genomics, except for this family here, which was sequenced using Illumina. And at this stage, we had no idea what caused the disease. There was only one gene that was clearly implicated in AHC, which is ATP1A2. But none of these people appear to have mutations in ATP1A2. So first thing we did here is we took these 18 sporadic cases and we uploaded them to Ingenuity. And here's the filter cascade that we used in Ingenuity. So just to go quickly through the filter cascade, you can see here that there were some 13 million variants in the set of 18 sporadic cases of AHC, affecting pretty much every single gene. So the first filter that we define is one to trim the number of variants according to frequency in the population. Since AHC is a pretty rare disease, again, it was very natural to assume that the variants causing it should be rare. So in this case, I chose to limit variants to those that are quite infrequent, 1% or less in the 3 different sets provided by Ingenuity. Then we definitely expect variants to be deleterious in some way. So in this case, I use pretty much default parameters provided by Ingenuity variant analysis. We decided to focus on variants that have good support. The default usually is 20, but in this case we prefer to go to 35 to make sure that we're not including errors in the analysis. Then comes the more interesting analysis, which is called the genetic analysis. So here we decided to go for dominant variants, de novo presumably, or very rare dominant variants in these families, so in these sporadic cases. And we focus here on those that are heterozygous or hemizygous, but we definitely wanted to exclude at this stage anything that was frequent enough to be homozygous. And then just to make it sure that we reduce the number of false positives, I added here a second genetic analysis filter that explicitly excludes variants that are homozygous or appear to be compound heterozygous. So, of course, we can also test those hypotheses separately, but in this case I was going for the clear dominant variants, and so these are the filters that made sense. And then finally, there is this filter for biological context, so to reduce the number of variants here from 46 down. And in this case, the logical thing to do was to say, I want to keep variants that affect or are known to affect alternating hemiplegia of childhood. Of course, since then, since doing this analysis and since the publication, the definition of what is known to affect alternating hemiplegia of childhood has changed. So if I were to reproduce exactly the same analysis as done back then, I would replace that with just affecting the gene ATP1A2. Since we expect new genes to be affected here, then it makes sense here to choose variants that are somewhat related here using this filter of up to 2 hops upstream of this gene. So using all this cascade of variants of filters, we could reduce the number of possibilities here from affecting every single gene down to affecting only 8 genes. So here are the results. And it was pretty much a slam dunk that you can see here, again, as expected, a gene like Titan gets false positives every time you look at it. As expected, again, there is a mucin gene as a false positive and a few other false positives that were not surprising. But then the important result here, of course, is the identification of ATP1A3 as mutated in pretty much every sporadic case. Okay, 15 out of 18, that's a pretty good result. And as you can see, some of these variants appear in more than one person, so they are not completely novel. And some other variants are completely private to one of the affected cases. So, to summarize that part of the analysis, as you can see, sometimes it can be pretty easy. So we start with all this large number of cases, and just by doing a single analysis, we can identify a very strong explanation for 15 out of 18. Sometimes it's easy, sometimes it's really easy. So in the case of this family that was sequenced by Illumina, I simply uploaded again the variants to Ingenuity variant analysis, and within minutes I had a very strong single candidate with mutations disrupting, very clearly disrupting a splice site of a gene that makes sense for AHC. So both parents here being heterozygous mutant, and the one unaffected child also being heterozygous. But then one of the 2 identical twins that was sequenced was homozygous for the splice variant, and that's a very strong hypothesis now to be tested. So sometimes it's really easy, sometimes it's a bit tricky. So going back to this family here, this large pedigree, we, after knowing that ATP1A3 is causing AHC in all the sporadic cases, we could recognize that it is also the cause, the very strong apparent cause in this family, except that in this affected uncle, there was a no call covering that gene. So that's why we couldn't recognize it earlier. So again, we can identify the causal variants for some of the sporadic, some of the families, but then we are stuck. Then we have 3 families here that we couldn't really interpret in any way. So we started doing everything we can to interpret these families, and it was simply one dead end after the other. And then it can even be confusing. So for example, when doing, again, trying Ingenuity analysis on this family here in the center, we can identify that the father carries a very strong and disruptive mutation modifying a glycine to a tryptophan, very highly conserved glycine. I mean, you cannot imagine a worse substitution than that. And then both daughters appear to be homozygous for the same variant, which clearly doesn't make sense. This is a completely novel disruption in the father. How come they have it homozygous in the daughters? So anyone who has done any analysis of genomes immediately should be thinking that maybe something is wrong here, that maybe there is a deletion, some kind of aneuploidy covering this gene. So this prompted me indeed to go ahead and try to understand and to look at the coverage over this gene to see if there is any evidence for a deletion. So just as a reminder that when doing resequencing, for example, as done by Complete Genomics, after extracting the DNA, there is a very lengthy sequencing process that produces a very large number of reads that are mapped to the genome. Bases are called based on that, and the final result is not only a series of base calls expressed as differences from the reference and no calls, there's also plenty of information about how much coverage there was throughout the genome. Now, Complete Genomics aims to provide an average of 50-fold coverage throughout the genome, but that is definitely not uniform. So just to give an idea of what that really looks like. So this is one arbitrarily chosen segment of the genome towards the end of chromosome 1. And you can see that the coverage is pretty far from being uniform. It goes up and down very fast. And I think it would be very reasonable for anyone to study a single genome and looking at the coverage profile like this to assume that maybe there is a deletion in this area, 20 kb deletion. But then if you study a second genome, completely unrelated genome, it looks like this. So here now the black line is the second genome, the blue one is the previous one, and you can see that both genomes appear to have a deletion in that area. Then you sequence another genome, and it looks like that. So after doing several genomes, you see that the profile of coverage is very uniform across the genomes, but not along the genome. And then again, you do get some signal that maybe some of the genomes do have a loss of material here, for example. If you do this for many genomes, in this case we went up to 500 genomes, then it's very easy to define what we call a median profile. So for every position along the genome, every 1 kb bin, for example, I obtain the median coverage level. And you can see again the dark line here that it is very, very noisy along the genome, but very consistent between different individuals. We can look not just at the median of the profiles, but every single decile from 0 to 500 genomes. So here you can see again a very confusing plot showing all the 10 deciles. And here you can see very clearly some individual that has a full deletion in this area. Still, it's pretty complicated, pretty hard to distinguish where the real deletions are. But by doing a very simple trick of dividing by the median, this plot, very confusing plot, transforms itself into something that's much more interpretable. So again, this is by very simply dividing by the median profile. And now we can see very clearly that most of the genomes have a certain distribution of coverage throughout this region. Some of the genomes here, at least 100 genomes, have hemizygous level of coverage, and then some genomes, at least one, has a full deletion in this area. So again, this now makes it very easy to identify here a 60 kb deletion in some genomes, and then there is also indication that some genomes have even smaller deletions that were previously completely lost in the noise. So going back to AHC, we could identify very clearly 33 kb deletion. And again, here you can see what the graph looks like for a small number of genomes before scaling to the median and after scaling to the median. And now this deletion is not just a random deletion. This is a deletion that pretty much explains this family here in the center. So now it makes sense. The father transmitted here a very disruptive variant. The mother has a deletion, so she is hemizygous over some part of this gene. And then the two daughters, both affected, inherited both bad variants. And they are essentially compound heterozygous between a very disruptive point mutation and the deletion. So this is a very nice hypothesis now explaining how the disease arises in this family. And then, now that we have this methodology for identifying deletions, and we actually have this specific deletion that we know about, then we can actually start looking at the other families. And to our surprise, we found that exactly the same deletion, which is not a private deletion apparently, it's rare but not private, is present also in this other family. In this case, both parents are hemizygous for that deletion and both unaffected. There's one lucky son who got both normal copies, and then both affected offspring here were less lucky and they got both deleted copies. So presumably no functional version of this gene. And then again, studying deletions genome-wide, we could identify yet another gene that was strongly affected by a deletion in this other family. So both the mother, which has AHC with somewhat different presentation, and the three affected siblings, all of them are hemizygous, deleted for this third gene. So to summarize, for AHC, we went from an initial situation with 5 families and 18 sporadic cases with no explanations, and now we have a whole series of pretty good, strong hypotheses for what is causing the disease in all these different families and sporadics. So towards summary here, what we are doing at ISB, we are applying multi-genome analysis now to a very large number of people, so over 900 people that we're studying in many different studies. And we are applying this multi-genome analysis to create improved statistical models throughout the genome. For example, here showing again the coverage analysis. And this gives us two different things. On one hand, it allows us to produce better filters. For example, the commonly mutated segments that allow us to reduce the number of false positives throughout the genome. And then on the other hand, it also allows us to understand the genome better, so the technology and the genome, and that reduces the number of false negatives. In other words, we find some things that we couldn't find otherwise. So to go back to the big question of how can we use information obtained by studying many genomes to improve the annotation of an individual genome. So again, by doing multi-genome analysis, we create enhanced models that not only tell us about the genome biology and teach us about the population genomics, but also gives us a very, very strong picture of technology biases and technology-specific errors. So using all those, we can significantly improve the annotation of every individual genome, even in the absence of a family, for example, to compare them. So I want to make one last point here about sharing information. So, for example, very recently, not to pick on that particular project, Neanderthal genome was released, and information was provided for the entire, all the reads in the genome. So if you want to obtain now the Neanderthal genome, you can simply go to the relevant FTP site, and you can download here a mere 160 gigabytes of data, which you can then process in your own computer at your leisure. And as you can imagine, this is very, very difficult for most people to do. It takes a long time, a lot of computation. So that's very good for researchers, but not exactly a convenient way to share data. On the other hand, if you go to Ingenuity variant analysis, you can simply select one genome and click Share, provide the email address of the person you want to share it with, and that's pretty much it. And you can also share here not just one genome, but a set of genomes, an analysis on a set of genomes. And this is indeed much more convenient way to share genomic data with other people. And of course, there is a small problem here. Genomic data is private, personal. So we have here an apparent contradiction that on one hand, we cannot really share genomic information with the public, with other researchers at large, because of privacy issues. On the other hand, we do need to share that genomic information. We need that for advancing science and medicine, as I was just describing. So all those multi-genome analyses would be significantly more powerful if we had access to the tens of thousands of genomes and exomes that have already been created worldwide. So that's an apparent contradiction, and I want to finish my talk here with some kind of a call for action. So we can find creative solutions to how to share the genomic information. So for example, if I now give you that coverage profile, that's significant information that is present in those personal genomes that is not personally identifying. And if I study a set of genomes and identify variants shared by many of them, then again, that's not personally identifying. So such a set of variants should be shared without any problems of privacy. So my call for action is, let's find new and creative solutions for how to share genetic variants, genetic information, without transgressing on the privacy of individuals. And let's just do that. Let's share the data as much as we can. So to finish, I want to acknowledge all the people that participated in this work, both at ISB and the University of Utah, and our collaborators in the University of Luxembourg, and collaborators from Ingenuity. And in particular, I want to thank all those people whose genomes were sequenced and we have been analyzing.
With that, I think we will end today's call. And again, I'd like to thank Gustavo and thank all of you. And we hope to see you all soon on a future Ingenuity webinar. Thanks very much.
Thank you.